exciting to be a part of the growth in the field of cryo-EM. So then in 2020, I went to start my postdoctoral work with Gabriel Lander. And we were focused on studying proteases and mitochondrial regulation using cryo-EM as well. And so I wanted to shift from studying these highways to starting to look at organelle crosstalk between these highways and then specific organelles focused on the mitochondria. But as you'll see in my talk today, I looked at HCV due to a funny story, a very interesting story that I'll map out in the next few slides. And so after my experience at Scripps with Gabe and also with the Andrew Ward lab, I was so excited to join the startup space here in San Francisco. So I worked at a AI first protein design company. And here is really where I got to explore studying programmable DNA integration, and in particular, large cargo insertion. And so that's something I'm continuing now at my group in the biotech startup that I'm now a part of. And we're still very, very early on in our growth, so still in stealth mode, but I hope there'll be opportunities to talk about this with all of you in the future, and very happy to answer more specific questions around that in the Q&A. And so, as Maria very nicely outlined, I was first looking at microtubule regulation, and what got me really passionate was intrinsically disordered proteins like tau that's involved in Alzheimer's and MAP7, how they're like these noodles until they're able to bind and interact with microtubules and form a very designated structure on the scaffold of the microtubule. And that led me to wanting to now look at mitochondrial protease proteins, because they are able to degrade misfolded proteins, and discerning between proteins that are misfolded versus not folded is a very particular challenge for these proteins. And they're really able to read shapes of both disordered and ordered proteins. And I found that biophysically very fascinating. But I actually joined the lab the very first day of the COVID lockdown. And that shifted my perspective in terms of wanting to get involved in however I could, helping to understand viral morphology. And so my neighbor, Alba, who actually had the same shift as me, where during COVID, you could only come in at certain shifts so that there were fewer people on campus. She happened to share that shift with me. And we really got to know each other during these times. And for me, it was my first few months. So she was one of the first friends I made, actually. And so she was working on HCV and actually said that it's a very flexible, heterogeneous, and largely disordered protein. And she was struggling to resolve it. And that got me so excited. And I said, hey, that's one of my specialties. That's one of my passions. I love looking at intrinsically disorders. How can I help? And I want to learn more about viruses because they're transforming my life as they do. And so that's really how I got to this hepatitis C virus project. And so for my talk today, I'm going to give you some background and outline some details around HCV structure, what was known about HCV, so the historical timeline, the scientific timeline, how did that get us to our core question of getting structural information about hepatitis C virus so that we can start to make steps or make critical steps towards creating a vaccine. And then I will walk you through the life cycle of hepatitis C virus. So this becomes really, there are a lot of moving parts here because this virus is quite complex, which I'll get into. So I'm going to give you all of that background and then showcase how we got the structure in my talk today. So hepatitis C virus is one of the most common bloodborne infections in the United States and is a major public health concern. So there are 50 million people with HCV worldwide, 1.5 million new cases appear every year. And there are almost 500,000 deaths every year related to HCV, but there are 0 preventative vaccines. And HCV can cause severe liver complications, such as liver cirrhosis or cancer, and more often than the other hepatitis C viral strains, so A and B, for example. And when we look at its anatomy, It's a positive single-stranded RNA virus. It has a lipid envelope and it has a viral capsid that encompasses the RNA. And then it has E1 and E2 glycoproteins. So when I was first learning about this, because I was so new to immunology, and there is a ton of jargon, like when I was first walking into it, I was like, whoa, there are a lot of heavy details here. And so a way I like to liken the glycoprotein, or the way I imagine it, is to the spike protein of COVID, and that forms a trimer. And so with E1 and E2 glycoproteins, these exposed proteins that interface with liver cells and initiate endocytosis and allow the cell to enter, these glycoproteins, no one knew how they folded in 3D. No one knew their shapes. So that was really what we were very excited to start to resolve. And so there are 8 HCV genotypes and 84 subtypes, making this one of the most diverse viruses ever, I think, after HIV. And it's a member of a larger family that includes yellow fever virus, West Nile virus, and dengue virus. And E1 and E2 are main targets for broadly neutralizing antibodies. And you can imagine these broadly neutralizing antibodies can come together and compose a cocktail in order to neutralize all of these different subtypes of the HCV virus. But there were very limited structural insights at the time. And what's really the history of HCV, or why don't we have a vaccine yet? So this was really surprising to me when I initially learned it, but the earliest description of HCV was actually found on Sumerian clay tables. So they thought it was caused by a demon, and the site of attack was the liver, which was believed to house the soul. Fast forward to the 8th century, they found that HCV was transmissible and contagious. And then super fast forward to the 1980s, there was the discovery that HCV is a collection of 10 proteins, a polyprotein. Then a crystal structure in the 90s resolved one of these components that acts as machinery for the replicon system. And then in 1999, the full replicon system was worked out. So the steps by which the virus is able to hijack the cell. And then there was a development of successful direct acting antiviral agents. And this was a huge discovery, and it led to the Nobel Prize in 2020. And so this was a major milestone. However, these antiviral agents are very expensive, quite inaccessible, and they do not protect against reinfection. So there is still a major need for a vaccine. But what's stopping us? So major challenges is that it is very heterogeneous, multiple subtypes, like I mentioned. Testing in monkeys has been very challenging. And because antivirals exist, it's often seen as maybe deprioritized by the larger community. But I feel that it is a priority. It's just very challenging. And because antivirals are an option, it's maybe harder to secure sustainable sort of funding and interest. But I do feel largely that this is a major goal. It's just very, very challenging. And then structural characterization of all the different conformational rearrangements associated with the HCV virus also remain largely challenging. So now I'm going to walk you through the key stages of the hepatitis C virus, or its life cycle. So HCV attacks liver cells or hepatocytes. And before it enters, it occupies this pre-fusion state. So in this state, HCV is loosely attaching to the surface and using low affinity Velcro-like interactions with different proteins on the surface. After this stage, you have receptor binding. So you have E1 and E2, that glycoprotein complex, which we know is a monomer, but we don't know its oligomeric state. So we know that E1 and E2, they form a heterodimer, but that E1 and E2 complex, that oligomeric state, whether it goes on to form additional units or come together in additional units, that's still an open question in the field. And so it binds a specific receptor, or CD81, a tetraspanin protein that's critical for entry. And this promotes endocytosis and membrane fusion. So it's taken up using clathrin-mediated endocytosis and engulfed by an endosome in the liver cell. And in this endosome, there's very low pH. And that low pH allows for a conformational rearrangement, that acidic environment. It changes E1 and E2, which puts E1 and E2 in a state in which it can initiate fusion between the viral and the endosomal membranes, releasing HCV's RNA into the cytoplasm of the cell. And this is really where the organelle hijacking can start to take place. So HCV exploits organelles. That's really critical here, and critical of viruses in general. But I'll get into what makes HCV so unique and part of why it's so challenging to study. So because it's a positive sense RNA, single-stranded RNA virus, it can be read by ribosomes as mRNA and then translated into one polyprotein that's cut up by proteases in the cytosol and or in the cytoplasm. And then it's cut up into 10 functional proteins. So these proteins include helices and RNA polymerases, and you really get the full network of machines to create and develop HCV particles, more and more viral particles. And so what's really interesting here that I was fascinated by was, so HCV makes these ER webs. So it's remodeling the ER's membrane and making membranous webs that specialize in replication compartments. So you can think of these as assembly factories. And this is really where you start to make all of those 10 components and polyproteins and be able to promote and create more and more HCV viruses. And HCV, what makes it unique here is that it forms lipoviral particles. So it mixes with triglycerides and machinery that's part of the secretory pathway, so EPO1 or B, and it essentially hides in plain sight as a fake, very low density lipoprotein particle. So essentially it's like a fat droplet, and it's able to get masked and hide in plain sight and get secreted. I'll just highlight this here. It gets secreted and then be in the bloodstream without signaling to our immune system that we have an invader, because it's hiding as if it's a fat droplet, which is really amazing that it can connect with these lipid particles, lipid-like particles, and lipoproteins, and cholesterol particles as well. So it makes this network to hide in plain sight. That is part of why it's so impressively able to reproduce and be so detrimental to the host. So that really puts everything into context for where the field was when I came to this project. So the pre-fusion state, there were components that were truncated components that had been resolved by crystallography. And so this was published in 2021 in Nature, very beautiful paper, where they have a portion of E2, and then they have it bound to CD81. So that was the receptor protein that I highlighted in the previous diagram. And so they were able to show that there's a large extracellular loop that maintains a closed state. And in E2, there is a CD81 binding loop that is in a retracted state. And what's interesting here is there are tyrosines that are essential for membrane binding that are positioned in their model very well, that if there is a change, these two tryptophan and this tyrosine, so these two residues are able to align themselves nicely in the lipid bilayer and help with that hydrophobic packing that needs to happen inside the lipid bilayer. So the structure showed a lot of promise for being physiologically relevant. And they also had A portion that they proposed was the receptor binding state. So they showed that in this state, this loop is now more engaged. So in pink is where the membrane would be positioned, and this is the exposed extracellular loop. And now it's in an open state. And so this loop from this binding loop from E2 would be able to engage with that open loop within CD81. So this was really exciting to start to unpack which parts of E2 are engaging with CD81 and elucidating that receptor binding mechanism. However, a major concern was the physiological relevance. So because E1 and E2 form a complex, looking at E2 without E1 is challenging because E1 might be affecting E2's conformation. So in a true physiological state, in the full length state, maybe you don't get these same conformational changes, and receptor binding isn't going to be as shown by the crystal structure. And so there are open questions for vaccine development and for structural biologists here. So could E1 binding affect E2's ability to perform those conformational changes that I highlighted? And then is E1 also positioned close to that membrane so that this conformational change can still take place? And E1 is the transmembrane part, or it's the membrane embedded part of the full E1 and E2 glycoprotein complex. So these are still major questions around the conformational rearrangements and the physiological relevance of those rearrangements. And then a question was, can E1 and E2 oligomerize? So can it form a dimer or a trimer? Again, these are really important when we start to think about designing mutations that can neutralize the virus in a physiologically relevant setting. So truly in the cell, and then when designing a vaccine, in the host. And then what interactions line the E1 and E2 dimer interface? So between E1 and E2, what's holding them together? It was thought to be mainly disulfide bonds, but there was really not enough information here to make concrete conclusions around what's holding this interface together. And then what role is that interface potentially playing in viral fusion? As we mentioned, the pH really affects how the conformational state of this E1 and E2 complex progresses. And so understanding how the two bind can be very important for starting to create hypotheses around viral fusion. So, and then finally, what broadly neutralizing antibodies bind the full length membrane embedded, so again, physiologically relevant state of the E1 and E2 glycoprotein complex. So our story begins in Amsterdam. So Quinten and Rogier Sanders in the Sanders lab at the University of Amsterdam, they worked extremely hard to identify which HCV strain and broadly neutralizing antibodies to visualize. This effort it took years. And so they were able to compare a lab strain with a clinically relevant strain, or a clinical strain from someone infected with HCV, that they screened against patient sera as well as broadly neutralizing antibodies. So what they saw was that this was indeed a diverse, clinically relevant strain because it required certain amounts of patient sera in order to get neutralized. So here, these values represent the serum dilutions and antibody concentrations in which HCV infectivity is inhibited, or essentially when is the virus getting neutralized. So for the broadly neutralizing antibodies, this clinically relevant strain required more antibodies to be neutralized, indicating it's very resistant. But for AR4A, which was really interesting, you see that it requires less than the lab strain, indicating that it may be very conserved in a way that hadn't been observed before. So the identification of this epitope for this antibody was novel, very, very exciting to see. And it it was a major step in indicating that we're ready for structural visualization of a full complex. So he worked to isolate the full length protein as well as each of these different antibodies. So he started with, he co-expressed with strep2 tagged AR4A with this Fab for it. And then that was bound with E1 and E2, the heterodimer, in order to purify the full length inside the membrane. And so using DDM as well to help stabilize the transmembrane domains. So what I really want to highlight to you here is how small E2 and E1 are. So E2 is 70 kilodaltons, while E1 is 23 kilodaltons, which for cryo-EM at the time, and I come from Gabe's lab where they've looked at even smaller proteins. So that was also, I think, why I was excited to take on this project, is that it was heterogeneous, intrinsically disordered, and so small. So there were a lot of interesting structural questions for me, and why I was excited to pursue and help out this project. And so Alba, who I mentioned earlier was my neighbor during COVID and brought me on and recruited me onto this project, she was able to get the full length structure with the final antibody. And this antibody was essential because it stabilizes E1. And the more antibodies bound, the more you get a picture of the epitope-paratope interactions. So she was able to stabilize it with peptidisc. And so for membrane biology here, the field was largely using nanodiscs and trying this with nanodiscs, but Alba thought very creatively to use peptidiscs or single peptides that line this very small transmembrane region of E1 very carefully, enough so that it's stabilized, but it also doesn't occlude or affect any relevant or interesting flexibility of the protein. So you're still getting a very nice physiologically relevant full-length structure. And now cryo-EM was possible. And so together we collected a 5,000 micrographs yielding 2 million particles. And so we plunge froze in liquid ethane using, I know this crowd can appreciate these details, using ultra gold 1.2, 1.3s, so 300 mesh holy grids. And then I made them hydrophilic in the Solaris for just 7 seconds. And so then we collected on the Arctica at a pixel size of 1.15. And so this was, using the Arctica. And so right away, what I noticed was the heterogeneity and the size. So I could see how challenging this project was going to be right away. So not just because it was small, and then not just because it was diverse, but really because this was a sample at the intersection of both of those very complex, very challenging problems for single particle EM. So we're looking at a 90 kilodalton structure here. And with the presence of the broadly neutralizing antibodies, it was up to 240. But the problem was that not all 3 antibodies were always bound. So sometimes you only had 2, and then we're really looking for situations where you had all 3. And in the situations where you only had 2, E1, the transmembrane domain was much fuzzier, so moving around much more flexible in those cases. So that was the broadly neutralizing antibody instability that added another layer of heterogeneity on top of HCV already having so many different subtypes here. And so what really worked for me when I committed to trying to resolve this very challenging project was using 3D variability analysis. And at the time, this was part of what it was getting released next to CryoDragon by the Zong lab, Ellen Zong. And so this was, but 3DVA was by CryoSpark. And so I was comparing and contrasting all of these exciting new tools coming out, but 3DVA, global 3DVA, turned out to be huge for me. And so what I played around with was clustering and separating the different conformational states. So this does a really good job of doing a lot of the work for you, especially now. I think the advancements that have happened since 2020 have been major. And I'm very excited to talk more about that in the Q&A as well. And so getting a chance to play with this tool as it came out was very exciting. And the developer, Ali at the time, was also very responsive. So if I had questions about how to use the clustering, how to set up different groups of trajectories and identifying novel trajectories, he was very responsive. And for me, my questions really lied around how do I mask out just the transmembrane domains? Because we're talking less than 20 kilodaltons, like really tiny, trying to visualize just a few helices, which people were telling me, you're crazy, you're not going to see that. And I was like, I want to try. And so local 3D variability analysis turned out to be a really powerful tool for just looking at the transmembrane domain. So of course we couldn't make out individual helices, but we could mask out regions in the transmembrane domain that when pieced together gave us enough of a full story. And so global plus focused 3D VA, or masking versus looking at the entire structure. So using a mask that had a radius of about 5 and a soft edge of about 5. So these are just different parameters you can play with in CryoSpark. So you could think of 5 as a threshold here. That depends on your size of your protein as well as the resolution that you're shooting for. And so this was really critical to get the final structure. And so what you can see from the selected classes that from about 1 million particles, and then this is just 2D classification in CryoSpark initially, you can see that there were features we could make out. And so when we started this process, we didn't know really what we were looking at. We know that antibodies tend to have this very traditional diamond-like shape, so like 2 V's coming together, heavy chain and light chain. And so we could make out the antibodies, but where E1 and E2 we're really trying to figure that out. And the antibodies, you don't know which one is which because they all have that similar diamond shape. And so I was able to isolate a class that had evidence of all three antibodies found. And that's really went into this global and focused 3D variability analysis. And so at this point, we were able to resolve the first full-length membrane-embedded structure of the E1 and E2 glycoprotein complex, bound to 3 novel, broadly neutralizing antibodies. So what I want you what I want to highlight here is you can see we're only able to resolve the transmembrane helices that sit right on the top of where they would be positioned in the DNA. There are lower resolution details that we tried to place in, but they weren't high enough to build a reliable model into. But what was nice is overall, we could get placement of the helices sort of right above the final 2 transmembrane helices. So a majority of E1, which was very exciting. And then we were also able to see sort of these green regions here. These are where glycosylation sites lie. And then on top of that, we could make out disulfide bonds as well, because we are able to get that resolution of around 3.5. And within the core of E1 and E2, the resolution was better. So that was really important for making out the epitope and paratope interactions for each of these antibodies. So the first thing we wanted to test was, how does our model compare to that previously crystallized truncated version of CD81 with E2? And so for the prefusion state, what we noticed right away is it aligned almost exactly. A few rotameric changes here, but very, very similar, giving us confidence that this has very strong physiological relevance. And it adopted that retracted conformation in the prefusion state. So that let us know that we have the prefusion state, which was great, because AR4A which binds in between E1 and E2, is supposed to lock it in its prefusion state. So really all the parts of our puzzle were coming together here. E1 also lied at a 20 angstrom distance, so sort of critical residues that have been tested or biochemically mutated and have implications in whether it or essential locations. So we were able to see that they were in the right spot, they were in the right location. So it was at the optimal distance to initiate membrane recognition for the membrane-associated helices in E1 and in E2. And so, yes, as I mentioned earlier, AR4A locks it in this prefusion state, which we hypothesize is metastable. So because of all that flexibility we saw, where you have AR4A kind of coming in and out of that binding state. So we're hypothesizing that it's quite metastable because of the flexibility that we saw. So an open question remaining is, will the full complex bind to CD81? Or will the full complex bound to its receptor protein of CD81 also reveal that CD81 is in that extended conformation? That's kind of the next step, is we have the prefusion full-length state, but the field would also want the post-fusion state. And that would be great to get bound to CD81. And then does CD81 binding initiate the destabilization of E1 and E2? So is that really does that binding event cause E1 and E2 to fall apart? But that would really be important to resolve in its full oligomeric state. So here we have E1 and E2, but E1 and E2 as a heterodimer then forms an oligomer that we weren't able to get at that time. And so what interactions were holding E1 and E2 together? So we observed hydrophobic patches to be I should say I observed hydrophobic patches that stabilized the sites, and the two complexes coming together and interacting with one another. So in E2, I saw that there was a hydrophobic patch here and along the stem, while E1 almost looked like a hand. So if you imagine E2 like a bouquet of flowers, this was the analogy we used in the paper. E1, the hydrophobic patch is matched where your fingers really would be if you were to wrap around a bouquet of flowers. So the two patches matched up really, really nicely. And this was the first time that hydrophobic patches were hypothesized to be stabilizing these two together. And then disulfide bonds we could resolve. And so the field largely thought that this was being held together by disulfide bonds. However, we see that the disulfide bonds were located or localized to within E1 and E2 separately. So they're critical for holding the two together, but not necessarily holding them, holding E1 and E2 to one another. So they're critical for stabilizing each domain, E1 and E2 individually, but not stitching them together. And this is important because earlier I spoke about how important pH levels are for changes along the HCV life cycle. So being able to have that mobility and change its location independently of disulfide bonds, or I should say change whether E1 and E2 are bound together independently of disulfide bonds, is interesting. And then whether E1 and E2 are stabilized individually, that being pH dependent is also interesting. So this was starting to elucidate the role that disulfide bonds, or it did elucidate the role that disulfide bonds play in holding and stabilizing the individual domains. We also saw the full glycan glycogens that were stabilizing the interface. And so we observed two glycans that stabilized the interface, which was very exciting to see, actually quite unexpected, and decorating one surface versus the other. So this provides really good evidence that you could have an oligomeric state, so either a dimer or a trimer, and you could have this oligomeric state where the glycosylated sides are exposed, but the non-glycosylated sides are all interacting potentially with one another, maybe through hydrophobic packing as well, or electrostatics and hydrogen bonding. And then finally, we were able to define how every single antibody bound in these individual epitopes. So for AT, let's see if my mouse, there we go. AT12009 binds at the head of E2. So potentially preventing E1 and E2's ability to enter the cell and get engulfed by the cell. Whereas AR4A, as we talked about earlier, is locking it in that metastable prefusion state. So trapping it in that state so that it can't separate, maybe later on during its life cycle. And then IGH505 targets a surface exposed alpha helix in E1. So it's kind of on the back in this picture, but this region here. And so, it's targeting a helix here that could be important, again, for making transmembrane interactions. So it could be blocking the ability to do that and to initiate fusion as well. Okay, and so here we have a summary slide just to summarize everything I've shown you today, which I know there's a lot of different components here. So to summarize, the E1 and E2 interface here is held together by an extensive hydrophobic cavity that here these individual pieces are really highlighting where these hydrophobic residues lie, and it's holding that cavity together. And then disulfide bonds are stabilizing the domains individually, but if you look at the interface, they're not. So we could see very clearly where the disulfide bonds are. And then the glycan shield or the sugar shield that helps the virus interact with those lipids and cholesterols that I talked about earlier and helps shield it from the cell. We're able to map out these as well. And so there's some additional glycosylation analysis that was also done by the Sanders lab and some more of his collaborators that did a really nice job going into the chemistry around each of these different types of glycosylation sites. And then finally, we get a summary of the epitopes. So where are each of these broadly neutralizing antibodies positioned, and how could that be preventing HCV from performing its viral properties? And so this is exciting to think about developing soluble immunogens and starting to map out vaccine design. And I know that is something that the Sanders lab is pursuing and very interested in, and that the field as a whole is starting to unpack. But the major questions remaining: can E1 and E2 play roles in viral fusion? And then what is the oligomeric state of E1 and E2? And that becomes very important for thinking about designing stabilizing mutations to stabilize that oligomeric state for further studies for vaccine development. Okay, and I actually just have one more slide that I wanted to mention as well. So this experience studying antibodies in particular was part of, I think, a huge reason why I was recruited to Profluent, where they were interested in designing novel enzymes in various areas. And my experience studying enzymes and the mechanics of enzymes and looking at visualizing these molecular machines, as well as exposure to the AI field, which I had never had before, is really why I'm excited to stay in the startup space and combine elements now from both of these experiences. And with that, I'll end here. I know we're at time, and I want to thank all of you for your engagement and attention, and as well as my PhD lab, my postdoc advisor, Gabe and Andrew Ward, and Alva, wonderful collaborator and COVID lunch friend there. And then finally, my experience at Profluent, and then my current role at the Cell Startup. So thank you all so much for your attention.
Amazing talk. Thank you, Lisa. Like really, really exciting work.
Oh, thank you. Thank you guys so much. I was like, oh, I'm I'm nervous there.
No, no, you did amazing, and it's really, really exciting.
Oh,
And I,
wow.
I do
Thank
have a
you.
question, but I'll just open the floor for
Yes, please.
other people to ask questions. So please, if you have any questions, raise your hand, or you can also type it in the chat, and I can help read it for you. Also, just a quick reminder that there is a mentoring session after this seminar. Thank you very much, Lisa, for um, accepting our
Oh,
invitation to stay
yes. Yes,
back.
absolutely. So we can go into more detail together.
Yeah, sure. Okay, so Meenakshi has a question. Meenakshi?
Hi, Lisa. Thanks, Myron. Hi, Lisa. Beautiful talk. Amazing
Oh, thank
structures.
you.
I was just wondering, you mentioned that there was a little bit of sample heterogeneity, like not most of the antibody particles you could see bind to the protein. So,
Yes.
apart from 3D variability and cryo-dragon, what do you think played a key role in helping you sorting out the best particles that would give to a high resolution structure?
Such a good question because I think the tools were one thing. That's always one element is the development of tools at the time. But another I would say was my experience with intrinsically disordered proteins before was that the more particles you have, the better. And then generally sorting, so using like ab initio and heterogeneous refinement, like sorting by at low resolution first, I found that to be really important to identify to help group uh, proteins or E1 and E2 that had three antibodies bound versus two. So in the beginning, separating those two was really important. And then using the model, the low resolution model that had three antibodies as a reference model to help collect and sort and go through that data iteratively was huge. So once you start getting to a good model, using that iteratively is really critical. And what I was concerned about there was bias, like maybe I'm biasing the which proteins are or which particles are coming out. But in this case, because it was so low resolution and we have very little structural information of how the uh, full E1 and E2 complex is formed, I was less worried about bias and more worried about getting enough particles because I, after I had a model around 4 angstroms that had three antibodies bound, I still had to do sub classification of the transmembrane domains. And for those, I actually grouped together samples from the three antibody group and the two antibody group because they generally had information about that transmembrane domain. But it would have been ideal to collect even more particles and do that iteratively again, which is of course a ton of work and very expensive. So I got to a point where the PIs were like, no more, you process what we have. And I was like, okay. But
Yeah,
very good
sure.
question. I think
Yeah.
in addition to the tools, um, understanding when and how to sort and collecting more particles was critical.
Definitely. I was also wondering if you use CryoSpark only for the processing or you would use other tools such as Reliant for the 3D refinement or something like that.
Yeah, great question. So Relion was more so helpful, I would say, post um, getting that 3.5 angstrom structure for like Bayesian polishing and trying to do CTF refinement as well. So that's upstream. So I think Relion still has some features that CryoSpark can improve on and really understanding how to use both tools. So similar, like I was talking about comparing cryo-dragon and 3DVA, because some tools are going to work better or worse for your individual particle set. And so really being able to play with both is critical. So I did definitely have to play with both. Yeah.
Beautiful. Thank you very much.
Yeah, great question.
Okay, we have another question. Vivek, do you want to unmute yourself?
Sure. Thanks so much. Terrific talk. I had a
Oh,
question.
thank you. Nice to
Um,
meet you.
Nice to meet you too. I had a question regarding AR4A, and that is that
Yeah.
it seems like, only a minority of patients end up at the solution of developing a broadly neutralizing antibody that stabilizes the pre-fusion state. Can you speculate based on the structure why this is such a hard solution for more patients to get to? What is it about the way the CDR3 loop is interacting, perhaps, or other structural features that's making this
Oh,
a hard solution
interesting.
to get to?
Yeah, I think because it binds both E1 and E2. So usually it will the other antibodies bound a single domain E1 and E2, but because there's so much motion at the interface, AR4A is interacting with both. I think that adds to the problem. But it's a question that I don't have a good answer for. I think it's something largely in the field that's not well understood. But oh yeah, did you have a follow-up?
Yeah, maybe just to follow. I mean, I think in Andrew's lab, you guys had solved some other structures of HIV antibodies and sort of had, with Bill Sheaf and such, start to understand how perhaps, from germline to mature, these things are evolving, and the loops are essentially affinity maturing and getting longer, and particularly for the HIV ones. Is there a similar thing going on, you think, here? Do you have any
Yeah,
other thoughts?
that's let me check the loop actually. I have the image pulled up here. So AR4A was interacting with CDR, yeah, H2 and H3. And what I see was that was the longest loop of all three as well. So maybe it had even more local flexibility as well because it's such a long flexible loop versus some of the others. That would be my sort of main takeaway is that
Okay.
it combined both and then potentially um, it has more local flexibility than the others. Because I see that it's the longest. I see that it's also the most has the most ring amino acids, so hydrophobic packing as well. And so maybe versus the others where there's salt bridges, I see disulfide bonds, you have this influx of low affinity interactions adding to that challenge.
Yeah, great. Thank you.
Yeah, yeah, no, but good question. I didn't give it a good answer.
Okay. So we have a question in the chat, and then we'll go to Josh.
Yeah.
The question in the chat from Nyman says, is there any tricks to capture the metastable protein at better resolution or increase its stability?
Yeah, so I know that while we were running into issues and when we were kind of in the hard part of the journey, the Sanders lab and Quinton tried to send us more antibodies. So that is an option where you try to stabilize with even more antibodies that are maybe interacting with other parts of the of E1 and E2. But as you saw, it seems like you're only having interactions on one side, where you're also having the majority of glycosylation sites, but you're not able to maybe fit another antibody. So the challenge there was like limited sort of real estate or surface area available to more antibodies. I would say like that's what comes to mind.
Okay. And now, Josh, do you want to mute yourself?
Yeah, thanks, Lisa. That was an amazing talk. And
Oh my gosh, thank you.
I worked on these proteins 10 years ago. I was doing crystallography, and it was very hard. So it's amazing to see this.
Oh, you so you know the struggle.
Yeah. This has been a structure that's sought out for decades, I know. So
Yes.
yeah, very exciting. And so I have a question about the flexibility of the proteins because I know that you've got these three antibodies to help lock down the protein's flexibility for the structural studies. But
Yes.
I'm guessing the flexibility is really important for their function. So have you or
Yeah.
others used these structures to maybe guide structure prediction or molecular dynamics to understand the flexibility and maybe how that affects oligomerization or fusion or things like that? Yeah.
Yeah, okay, there's three reasons I love this question so much. So, the first is that we did try molecular dynamics, but we couldn't make out anything significant, especially looking at RMSD fluctuations between the three antibody state, two antibody state, and then empty antibody, like our APO state. So we didn't include that anywhere in the paper, but I personally, so at the time we were publishing this, it was right when AlphaFold was released. So you can imagine the blood leaving, our face, the fear that they're going to be able to predict this complex and there's no need for us to do this anymore. And the truth is it could predict really well the two individually, but not the interface. So I think for flexibility studies, I know that I think that revisiting molecular dynamics today would actually be really interesting because there's been so many advances in the tools and doing local molecular dynamics. So we were trying to look at overall the entire landscape, but I think looking at individual regions and sort of applying that same masking technique that I use to resolve the structure piece by piece, I think looking at them individually, masking out their local fluctuations could be much more effective.
Yeah, that's really cool. Do you mind if I ask another question, Maria? Is that okay?
Yes, please.
All
Sorry. The glycan stabilizing the dimer is really unusual. And
Yeah. Yes.
have you? I mean, I'm not sure if you've seen this in other viruses. Have you? Is it something that you think maybe has been overlooked?
Yeah, so I love, okay. I only talked about one of the reasons I love that first question, but this question also, when one of the reviewers brought this up, that he had never seen this before, and the question was why, or why are those two in particular unique? So the Sanders lab that worked with they worked with chemists locally at the University of Amsterdam, another group, to try to analyze each site and identify why these might be unique. And so again, there wasn't anything significant about these glycosylation sites, like comparatively to the others, that gave any real, like anything we could use, anything conclusive for the studies. But structurally, what I see is that they had some of the longest branches. So that literally just gave them more real estate to access both E1 and E2. And then where they're sterically the largest, so where the rings are, is further away. So the individual branch is longer and thinner, which maybe allows for flexibility across E1 and E2. But I'm completely speculating here. It's a very important question that I don't know who's working on this. I should revisit this one day.
All right, thanks, Lisa.
Yeah, thank you.
Okay, before we move to Lucas' question, because it's already 11:03, I'll just like sort of officially close the seminar, but we know that Lisa is staying behind anyways, so we can keep asking questions, and we have the mentoring session. So I think it was an amazing talk to close this year's seminar series. Thank you very much to everyone who joined today. Thanks very much, Lisa, for sharing with us these amazing outcomes from your research. So see you all next year. And now moving to Lucas' question.
Hi, Lisa. How's it going?
Hi, good. Thank you so much.
Yeah, nice to see you. My question is, I wondered if you'd looked at sequence conservation across different viral strains and whether you could map that back onto your structure. For example, like
Yes.
were the predicted sites of oligomerization highly conserved or
Yes. So we do have a figure in the supplemental. Let me see if I can pull it up and share. Well, we saw that the interface between E1 and E2 was very highly conserved, and the part where it's interacting with AR4A, super highly conserved. But then the regions around E2's head, and then right where the transmembrane domains start, lots of flexibility. But let me see. Let me download it because we have time, and I can pull that up. It's a great question.
Nice.
An important question. Yes. So let me see. Yes. Okay, I'm going to share my screen again. Also, you guys, you got to ask Vivek to send me his question so I can ask Alba, because it's going to eat at me later on. Okay, let me share this. Let me share my full screen. Let's see. Can you see my screen now? Oh, wait, no, wait 1 second. Okay, can you guys see this PDF?
Yes.
Yes. Yeah.
Okay, great. So, where the hydrophobic regions were, you can see like in E2, very, very conserved. And then E1 as well, the hydrophobic patches, super conserved. But then you had regions in E1, not hydrophobically, not hydrophobic, not conserved, or conserved but not at a hydrophobic patch area. But then it would be like you see a salt bridge. So that was really interesting to map out. So we went through every single interaction. So the reviewers asked for this, and we had done it more generally, but we went through every interaction for the strain that we had, the AMS strain and the H77 strain. But I think it could be interesting. Oh wait, let's see. Actually, I don't know the exact number of the total strains that we modeled it against. So I want to say it was at least 1,000, but that was done by Rohir's lab as well. Like a ton of different strains were all in this comparative analysis.
Yeah,
but I
nice.
don't know the exact number. But we went residue by residue to compare conserved versus individually hydrophobic residue. So yeah, great point. And then this point, or this region was like numerically probably one of the most conserved patches, and it has hydrophobic pocket, and you can see salt bridge activity as well. So that's interesting, like where you have more than just one group of important interactions, the conservation was higher, and very much so at the interface. So here the structure is E2, and then E1 is kind of, I don't know, can you guys see my hands?
Yes. Yeah.
Oh yeah. Of course you can. I'm sorry. So it's really where that line, where this purple region is where like the palm of the hand would be in the bouquet, and then this region is where the hand would be touching the bouquet. So I have to show you guys quickly. Just the naming for this was so funny because we were trying to find a way to describe this. So this is Alba's hand that I drew around to try to explain that it just how the patches wrap around the stem. So we called it stem in hand model, and then we got to name all the individual parts because we're seeing them for the first time and how they fold, which was exciting. But this was very contentious. The reviewers almost were like, no, you can't put that in there. But I was like, I demand it. But sorry, that's a side that I digress. But yeah. So very good question. The conservation was mapped out residue by residue.
Excellent. Thank you.
Yes.
Cool. I think we're officially in the mentoring session of the seminar, I guess.
Amazing.
Cool.
There's still so many people. I love it.
That's great.